Hello everybody, this is my exam 3 write-on. My name is Cash Merrill and I'm going to be going through the problems with you for the write-on for exam 3. So let's go ahead and get started. Number 1 is talking about specific human diseases, and it gives us a list of terms here, and it wants to know which of these terms is associated with these diseases. So Patau syndrome is the first one. I'm not really sure how to say it, but it's when there is an extra copy of chromosome 13, I believe it was 13. And so this happens during meiosis when there's a non-disjunction and it creates trisomy, which is you can also call aneuploidy. Aneuploidy is any time when there's not the correct number of chromosomes. So trisomy, aneuploidy, and non-disjunction is for part A. Part B talks about conventional form of Down syndrome, which is trisomy 21. So same thing here. It happens during meiosis with non-disjunction. You get an extra, two of them don't separate, so you get an extra copy of chromosome 21. So it's trisomy, aneuploidy, and non-disjunction. Letter C is talking about Turner syndrome. So Turner syndrome is when females don't have one of their X chromosomes, they're missing an X chromosome. And that's also during meiosis, it's just one of them gets caught and they don't end up with two of them. So they got non-disjunction, monosomy, aneuploidy, and non-disjunction for Turner syndrome. Part D is Cry-du-Chat syndrome, which is a terminal deletion on chromosome 5. And when you have a chromosome deletion, you still have the same number of chromosomes, but you're just missing some important information on that chromosome. So it's just deletion is the only one that works for this Cry-du-Chat. And letter E is type A hemophilia, which is an inversion in the X chromosome. So the only one for this one is inversion for type A for letter E. So that's number 1. Let's go ahead and move on to number 2. Number 2 is talking about an E. coli DNA sequence, and we're going to talk about replication. So it gives us 2 primers here, primer A, primer B, and it talks about the DNA lagging strand here. It gives us that. And the reason we know it's a DNA lagging strand is because it says this will be involved in the newly synthesized DNA will be synthesized as Okazaki fragments, so which deals with the lagging strand of the DNA. So I wrote that out here. It's kind of long, so I had to make it into 2 lines here. So that's just a continuation there. So for part A, it's asking what is the polarity of the 2 primers, indicate the polarity. Well, since this is the 3' end, so I went through and found where these primers matched up. So C, G, T, A, U, C, G, so forth. So I found that primer A matches up here and primer B matches up here. And so since they match up in this order, so A, U, T, A, C, G, so they match up in this order. So since this is the 3' end, this has to be the 5' end, and this has to be the 3' end. Same with primer B, 5' end, 3' end. So the 5' is on the left, 3' is on the right there. So letter B wants to know what is the DNA sequence that will be synthesized in the middle of the 2 primers. So we have primer B and primer A here, and the sequence in the middle, so it's going to be synthesized from 5' to 3', so this way to here. So following along here, I have, so starting with the 5' end, I got C, G, A, G, C, T, A, C, T, C, C, A, G, G, C, A, C, G, C, 3'. So that's the sequence of DNA that is synthesized in the middle of the 2 primers. Letter C wants to know which direction is the replication fork moving as this daughter strand is being synthesized. So for this, you got to know, so if this DNA represents the lagging strand, there's a DNA sequence on top of it that represents the leading strand, and the leading strand will be opposite, so it'll be 5' to 3' here. And so since the DNA is going to be synthesized from 5' to 3', the leading strand will be synthesized in this direction, and the lagging strand is synthesized, so let's say, so this primer B was put on, boom. So now you have a 3' end here, and it's going to be synthesized 5' to 3' until it hits the next primer that's down that way. And then primer A gets put on, boom, now you have a 3' end to work with. And you get synthesized here all the way up to primer B. And then another primer is going to be put on, boom, another primer, boom. So it keeps going this direction. So the replication fork is moving then right to left. So that's the answer for letter C. The replication fork is moving from right to left. Letter D, which primer was put on first? I just kind of talked about that. Primer B is put on first because it's got to get put on, then synthesized up to that primer. Then primer A is put on, synthesized up to primer B. That's what makes the Okazaki fragments. So primer B was synthesized first. Now let's move on to number 3. So number 3 is talking about an inverted chromosome. So talking about a chromosome and that gets inverted, but still going to try and synapse during prophase 1 with the other chromosome, so that they can then segregate into their appropriate gametes during meiosis. So the first chromosome we have is normal. It's just L, all capitals, L through X, with centromere between N and O. And then the next chromosome talks about is the inverted chromosome, which is LMN, centromere OPQR, WVUTSX. So the inversion here happens between W and S, so it should be like this, but it just gets inverted right here, so it forms this little loop. So during S phase, both these chromosomes got replicated, and then during prophase 1, they try to line up here. So in order to line up with this one, it has to form this little loop so the base pairs can line up, so it can synapse during prophase 1. And then it's telling us that there's going to be a crossover occurring in prophase during T and U right here. So then it wants us to indicate the 4 products of meiosis after this crossover. So to find the 4 products of meiosis, each of these chromosomes is going to separate into its own gamete, correct, during meiosis. So first we can look at our top one here, which is just going to be our normal chromosome. So it's going to be capital LMN, centromere OPQRSTUVWX. And that is our normal healthy chromosome, so it's normal and healthy. The next product of meiosis, we're going to look at this strand right here, which does not have any crossing over occurring either. So it's just our original inversion chromosome. So it's lowercase LMN, centromere OPQRWVUTSX. So it's our inverted chromosome, and it's also healthy. It's fine. Our next product of meiosis, we're going to look at this strand right here. So it's capital L, capital M, N, centromere OPQRST. Then after T, it's going to cross over to the lower cases now, U, V, W, R, Q, P, O, centromere again, and ML. So this third product of meiosis has 2 centromeres. So it is our dicentric fragment, and it's an unhealthy chromosome. And the fourth product of meiosis, we're going to look at this strand right here, which also undergoes, well, it's a formation of both of them, like the first one. So let's start right here. So we have capital X, capital W, capital V, capital U. Then we cross over to our lowercase t, lowercase s, lowercase x. So this one right down here, you can see, has no centromeres, so it's acentric and unhealthy. So those are our 4 products of meiosis there. And that's number 3. So now let's move on to the last problem, number 4. Number 4 is also talking about an E. coli gene, a DNA sequence, and we're going to be talking about transcription this time. So it gives us this DNA sequence, and I've indicated the +1 site here in bold on this gene. And it's telling us in the problem that this gene is transcribed from right to left. So from here down that way is being transcribed. And then letter A is asking us which strand is the template strand, which strand is the coding strand. So since we know that transcription is occurring from right to left, we know that transcription, the polymerase reads the template strand 3' to 5' and synthesizes the mRNA strand 5' to 3'. So reads it 3' to 5' and creates it 5' to 3'. So that means this upper strand must be the template strand, and this bottom strand, the lower strand, is the coding strand. And another thing that tipped me off there was that in the instructor notes it said that the coding strand, the +1 side, is often an adenine nucleotide. So we got A there. So coding strand, upper is the template and lower is the coding. Letter B wants to know what is the mRNA sequence that will be produced from this DNA molecule. So we look at our template strand. When we start at the +1 site, the +1 site is indicative of the first nucleotide that is transcribed. So we're starting here with T and going down that way, and this is going to be our 5' end right here. So going from the +1 site down that way, it'll be 5' A, G, C, C, U, G, A, G, U, A, U, G, 3 prime. So that's the mRNA sequence that will be synthesized from this gene starting at the +1 site. Letter C says in which direction, left or right, along the coding strand relative to the +1 site, would the -10 and -35 sequences be located? So the -10 and -35 sequences, since we're starting replication here and going right to left, or transcription, excuse me, and going right to left, the -10 sequence is known as the TATA box, which you can actually see right here, TATA. And then the -35 sequences would be farther down this way, but they need to be before the +1 site. So the sigma factor recognizes them, and then it guides the RNA polymerase to start replication, the correct spot. So I said that the -10, -35 sequences are located to the right of the +1 site on the coding strand. And letter D, it's asking us where would the terminator sequence be located relative to the +1 site. The terminator sequence is where it tells to stop transcription. So it needs to be towards the end, so it's going to be on the left of this +1 site. So the terminator sequence is on the left. So those are the problems for the exam 3 write-on, and thank you for watching.